the Agrobacterium mediated genetic transformation in plants. Transgenic plants are the plants that are developed by transferring genes from one plant to another plant. We can take genes from any source, from plants, from animals, and as well as from bacteria. The plants are the transgenic plants developed that are resistant to various biotic and abiotic stresses, extreme environmental conditions, and also there are certain transgenic plants that develop and show resistance against a particular herbicide, weedicide, etc. Apart from this, there are certain other plants, transgenic plants, which develop by transferring animal gene, and they are used to produce animal therapeutic proteins used for the human therapy, like edible vaccines are produced in transgenic plants, interferon, interleukins, antibodies, antibody fragments. All these can be produced successfully in plants, and their application is so good that they provide immediate immune response, and they are used for the treatment of various diseases. This Agrobacterium is a soil bacterium that is gram-negative rod, and this bacterium is not naturally found in the soil, and it is a natural gene transfer mechanism exists in this bacteria. It transfers the gene into the plant, but it is a pathogen causing crown gall disease in plants, and genetic engineer utilize this techniques to transfer the gene of our interest. This Agrobacterium contains tDNA. This tDNA is actually the transfer DNA that transferred from bacterium to the plant, and it is actually infected at the wounding site, which is present in the collar region. Collar region is the region where the root and stem unite, and when it enters through the wound site, it produces a tumor-like outgrowth, which is known as gall formation. And because it is in the crown region, it is called crown gall. And in crown gall, bacteria produce opine. Opine is a special class of compound that is produced by the bacteria and utilized by the bacteria, but it is not utilized by the plant. So it is waste product for the plant. But most of the protein of the plant is used for the formation of this type of opine, which is useful only for bacteria. So bacteria produce this tongue, ultimately the yield of the plant is reduced. It is a disease, but this property of this Agrobacterium is utilized for the gene transformation in plants. Next slide. In this slide, I am discussing about how gene transfer process takes place. The bacteria contain this Ti plasmid. Ti plasmid means tumor-inducing plasmid. This contains an origin of replication, which is autonomous in replication. This opine utilization gene. The bacteria produce opine, and it also has a opine utilization gene which utilize opine and live on that opine. There are so many opines: succinopine, manopine, argopine, and several others. And it is based on the strain, which strain of Agrobacterium is infected. This Ti plasmid contains tDNA. This tDNA is actually transferred, and various proteins help. The helping proteins are encoded by Vir region. This is composed of many genes. Vir region contains Virg genes, which is actually a sensor. It senses the plant secondary metabolites. This is the plant signal, and secreted by the plants. Now these signals are sensed by the Virg protein, and they transduce the signal to transfer the tDNA. Now the tDNA transfer process starts. There is a B2 protein which protects the DNA, and some endonucleases cut the DNA from that side with the left border and right border and transferred into the plant cell. This VD4 and VD protein produce a channel for the transfer, and this E2 protein is a single-stranded binding protein which binds the DNA and remove the single stranded DNA cannot form the secondary structure in the presence of this PE2 protein. Now it is transferred and integrated randomly into the plant genome.